talked about it in Now's talk as well. So we are now going to talk about visual awareness as in qualia. A little difficult to define. Phenomenal experience, to feel, to be you, yeah, it's just a feeling of perceiving something. Has anybody a better definition than I do have now? Now you have written the reviews about qualia. A better one?
Sense of presence.
Sense of presence?
Yeah.
I don't know. Some people claim that the sense of self is necessary, but I'm not so sure about that. Is that what you mean by?
No, not self. I mean, you can get lost in music or in walking or whatever,
right.
and really de-emphasize yourself. That's often a very relaxing feeling. So presence, very hard. It's sort of like almost atomic. Being there.
Being there.
The word being is very hard to define. Since every definition is something. So how do you define is?
Yeah. So I guess for the moment we have to pretend that we know what we are talking about. So I guess just look at a blue dot and just experience it, and that's sort of what I mean. So here are our consciousness zombies. So, and that's the problem, right? So if I would want to know what you are seeing, and I want to study now your perception of this blue dot scientifically, then I sort of need to trust you on that, right? Because theoretically, I mean, you need to tell me first, because it is a subjective experience, right? I cannot really access it. But theoretically, you could actually see red and call that blue, or you actually, the other way, you say blue, but indeed you would perceive what I would call a red. So there is not much we can do about it. Or you're a complete zombie and you just tell me I see a blue dot and you actually don't see anything. So that is the presumed computer state. So we may argue about that as well. But yeah, so that is the problem. And that is indeed the fundamental problem. This is also why we had organized the symposium at the consciousness conference about the no-report paradigms in consciousness research, because we sort of collectively realized that we have a little trouble to study the neuronal correlates of consciousness, because we always have the behavioral indication in between. So what we really want to know is the correlation between the awareness state and the brain state. And then we have the behavioral indication of the subject in between, and we have some good or, I mean, worse or better measure of brain activity. There are multiple measures of brain activity. There are, they have different temporal scales, different spatial scales. They are reliable, non-reliable. Some of our neuronal correlates are even different for the different brain measurements. So yeah, so it's really here the imperfect science comes in. But we still want actually interested in this here. The way we approach this problem, because we don't know what the best signal is, and to some extent we don't even know what the correct level is, we in the lab sort of try to look at different spatial and temporal scales. In human studies, you can use, for example, an MRI scanner. So this is the mostly used method together with EEG to look at ongoing brain activity while somebody is perceiving something. And then at the same time, we try to get to the cells. And this is sort of, cut a little movie for you. To make, so theoretically, with real-time MRI, you can really see what's happening in the brain. in terms of bolt activity, but bolt activity is very coarse. And we still think we can debate that, but that the neuronal correlate of consciousness will be in the neurons that are firing. And in order to get to the neurons, we do cell recordings, and this is why we actually take monkeys instead of humans. This is an MRI call, and this is our setup, just to give you some feeling for how this type of experiment would look like. So the monkey is trained to perform a given task, and he sits there in a head-posted fashion, and he is trained very long, sometimes like 1-2 years, to really perform those tasks. But the really nice thing is that we can really put an electrode in the brain and really measure cells. So there are different scales at which the neuronal correlates of consciousness can be accessed. Mm-hmm.
What's the current spatial scale of MRI? How many neurons?
A couple of millimeters. It's
but how many neurons in a
milliards.
Okay, so
yeah, so it's
we're not even close.
Out of, yeah. Exactly. So that's actually why I showed you the movie. It's really one voxel just contains. Yeah. Exactly. So yeah. So we have the behavior. We have whole brain regions, and this is what most functional MRI scientists would use. They talk about brain regions, in the worst case, even about lobes. I'm going to get to this point later, but the parietal lobe, for example, it's crazy. I mean, but so. And then there are neuronal populations. You get to them, for example, with imaging methods, like two-photon imaging. This is a very cool technique. Unfortunately, it doesn't work in a monkey. I would love to have that. You can really see cells you are recording from. This works, for example, with calcium indicators, and it's very cool. So you can really see, with the calcium indicator, you can really see when a cell is firing, because like it really becomes brighter or less bright. And you see the whole network at the same time, and you can label different cell types. So it's really the dream of the neuroscientist. The old-fashioned way, and that's the working and also very useful, is to put an electrode into the brain and measure extracellular potentials. And from that, we can infer to the spiking of pyramidal cells. There is a sort of a selection bias towards big pyramidal cells, so we don't get interneurons that much, and we also don't get astrocytes, for example, because they are not electrically excitable. So they may be very important, and we just miss them. So in humans, we have the function MRI. Also, lesion studies are still very helpful, because if you have some sort of double dissociation, I think that's the gold standard in cognitive sciences in general. If one function is still working, the other one is not working, then you can at least say, okay, this brain region seems to be important for a given function. And by now, there are also many non-invasive stimulation techniques, like transcranial magnetic stimulation, where you can influence electrical activity in the brain by basically inducing current by a magnetic coil. And there are also techniques that are called like transcranial direct and transcranial alternating current stimulation. There have been also, there was just a study from Stephen Lorries who showed also improvement of cognitive functions in vegetative state patients with transcranial direct current stimulation. Super easy technique. It's sort of really just 2 electrodes here and there, say like this, and then you just run a current through. The problem is the effect sizes are very small. So we are playing with that also a lot in the lab, and it's sort of cool because you can apply it also in a frequency-specific way and all that, but I'm not so excited about effect sizes. So, but yeah. So now if you want to sort of, I guess we are all interested to understand consciousness in humans, or at least I am, if you're not interested in invertebrate consciousness, certainly also an interesting topic, but at the end of the day, I think it's about us. And in order to make this link back, we sort of need to use the same measurement technique. So this is why the monkey fMRI is also very important, because it sort of links it to the human imaging. And in monkeys, we can go many steps further, which has been very useful, for example, is to do like just local inactivations. Optogenetic manipulations are also very nice. And we can do in the monkey cell microsimulation. Basically, we can do the full workup, only to photon imaging, like really looking at the same time at the full network. That's what's not working yet. Is what I would like to have, is what people can do in the mouse. One red. Yes.
Practical question. What's kind of the turnaround in optogenetics or something like that? How long does it actually take to isolate the thing, genetically?
Yes. I think like in the large monkeys, like the rhesus macaques, so there are no transgenic rhesus macaques, right? There are some Japanese transgenic marmosets, but there is no line that you could use now for optogenetics. So you cannot just buy them like the mice. So they would not express the receptor. So you need to inject the virus. Then you wait like a month or 6 weeks until the gene is expressed, and then you sort of get lucky or unlucky, you kill the cells, or you didn't. So it's sort of risky, especially if you have a trained animal. And the disappointing thing about the optogenetic studies in the rhesus macaques, and that's also precisely the reason why I'm still going with pharmacological inactivation and electrical microstimulation, because there's sort of no real behavioral effect of it. So there have been multiple studies who have performed optogenetic stimulations, and the most you get when you are really in a small structure, like the superior colliculus, is some change of eye movement latencies or so, but it's like on the level of 3 milliseconds. So it's really, it's not very useful. I do have more hopes for the marmosets, because they have a smaller brain, and in them it may actually work better than in the mouse and in the rat. There's also, for some strange reason, virus expression is also not so good in the monkey in general. So there must be also some immune response related things. But obviously it would be the way to go, because the cool thing about the optogenetic is that as long as, if you know the promoter, and for the monkeys we also don't know that. So then you can sort of express your receptor only in the cells that are of the type you want to study, like for example the astrocytes or a specific type of interneuron or so. But this means you need to know the promoter, which we don't know for the monkey. This is also why I think right now it's not there, but I have a huge hope in the Japanese neuroscientists working with the marmosets to actually get that to this point. Is somebody, I heard from now yesterday that somebody is doing marmoset studies here? Yes? Okay, cool. I'm going to talk to you. What's your take?
Yeah, so just I do with getting a fix. Yeah, now I don't know what. Yeah, sorry. Yeah, just I do is getting a macroconnect. And so I want to discuss about the sense of the marmoset after.
Yes. Okay, good. Okay, so this is the study question. What do you think is the correct spatial temporal scale to actually look for the neuronal correlate of consciousness? I put throughout the talk, I put a couple of questions that I sort of, you can pick, but this is one I'm very interested myself. The monkey training, do you want to see that? You don't? That's important. Okay, so in order to study visual awareness, we had it yesterday in Nao's talk already, we sort of want to make sure that the visual input remains the same, and like the basic visual processing is the same, and the only thing that really changes is your perception. So then we have at least one confound factor we have already sort of discarded. And in order to do that, the ambiguous or bistable stimuli are very handy. So this is one of them. Who sees it left? Okay, who sees it right? Yes, and then it switches, right? And the nice thing is it's really, there's no change in the physical stimulus, but it's really just changing in your perception. Okay, so that's one of the stimuli that is very handy to use. Yeah.
Is there any trick to making it change? Because
Yeah.
I can't make it change.
Yeah, you could make it change, or you cannot.
I can't.
Yeah, it's. I think it's very okay. Yes, you can, for example, blink like this, but it's more a probabilistic thing. So it's not that you blink and then it changes, but you can sort of do like this, and then it increases the probability. And you can also concentrate on one versus the other, and then you can shift it a little. But it's always probabilistic, so it's not that you can say, okay, now I make it change, or now I just make it look left or right.
I think if you add a bit more action, so if you just try to swipe it with your head, you can actually just see it make left and right, whatever you want.
That's a cool idea. It has been done by Randolph Blake's laboratory. But I think the effect size was also very small. Yes?
Yes, well, yeah, many people who believe that action is really important tried it a lot. I'd say the effect size is relatively small. But that fixation location definitely affects, because that directly affects visual image.
Yeah.
So if you look to the left edge or right edge, it probably changes. Mm-hmm. Right? Yeah.
It's very easy for me to switch. I switched.
Yeah.
I switched.
How do you do that?
How you do
never
it?
switches.
You fix one point and
Yeah.
you follow it and you think
Yeah.
yourself
Front or
behind
back.
or in front.
Okay, yes. Okay. Yeah, obviously in the lab, yeah, with every electrophysiology experiment we would do, there would be always a fixation spot, also in a human psychophysical experiment, so you would actually check that there is no eye movement, but yeah.
Are you going to talk about the individual difference on this?
No, I'm not. Go ahead.
There is no huge individual variance on switch rate. Like some people switch really rarely, and some people switch very fast, and some people claim that it is related to schizotypy or the distractibility, and there are some volumetric study that correlates the thickness of the parietal cortex or frontal cortex to be related to individual variance. It's really, but it varies a lot.
I can tell you how you can stop it by just removing it intermittently. It was like the first scientific study I've ever done, and it's very funny because you just remove it for a couple of seconds and then you put it again. And if you do that, then it can really get stuck like 4 or 5 minutes, always in the same state before you removed it. So it really works.
So can you fix yourself in a state where you see it as both?
Yeah, this happens. Yeah, so that this is one of the disadvantages of this stimulus, that when you really look for a long time, like then sometimes you cannot tell anymore. But obviously in the lab, our stimulus is a little better and it's a little faster, not so jerky.
You switched.
Okay.
Wow. I wonder whether it makes a difference if you're in front or from an angle. I couldn't switch it sitting here, but sitting there it was relatively easy.
Okay.
Do you know whether
Right.
looking from the side makes a difference?
I
or
only look
maybe
from
I
in
was
front.
just getting used to it more, I don't know.
I'm not aware of the effect of the viewing angle, but there is some kind of physical interpretation of the thing as well.
Mm-hmm.
Like if you put two things at the same time, you tend to see either this rotation or this rotation. Because it is physically possible, right? If there's a friction. And so if you see it from angle, maybe it's hard to interpret one direction or the other. I don't know.
It may just be chance that I kept trying. And,
Maybe it's the change. Maybe it's the change.
yeah, it could be, yeah.
Kind
So
of like you said.
that's why I have to do many experiments to check. We should probably
Check
wind up, and then after the break you can continue.
Okay, let me actually see whether. Yeah, actually, yes, that is a good spot. I just have to hurry up after the break a little more. But it's good. So after the break, we are going to talk just about conscious visibility.
But I would like to read the response of Nico Schiff,
Okay,
real time.
good.
I sent him an email, and his answer is to the question which I mentioned about this patient who said that he was in pain and that he got surgery. This case is one of those reported in Forgax, is that how you pronounce it? Forgax et al. 2014, but I cannot recall if the anecdote is in the supplementary history or not. We also did a presentation at Society for Neuroscience last year, just on communicating this case, Mendels et al. 2014. I will send a poster tomorrow. Basically, all such patients are misdiagnosed as vegetative and have very preserved thalamocortical function. In this case, very severe brainstem injury led to basically no oculomotor or motor function at all, including no blink or auditory localization responses. Really an extreme situation. So this answers your question. Definitely okay to mention the detail. I asked whether I could mention this in the group. Communication channel was discovered roughly 3.5 years after injury. Gallbladder pain was identified in first 24 hours of using it, and next day evaluation showed it was acute and he went straight to surgery. No joke, no exaggeration.
Wow.
So,
Okay, so that's good, so it's true.
So now we can have our coffee without uncertainty.
Half an hour, right?
Yeah.